So let's talk about different types of mutations. And so the example that I gave you with hemoglobin and sickle cell anemia, we're actually going to investigate that a little bit further in lab next week. And we're going to talk about what is referred to as a point mutation. So a point mutation is where you simply have a substitution of a base, of a single base. And some chemicals can cause point mutations, and so can ultraviolet radiation can lead to point mutations. So when we look at an example, we could have a DNA molecule that says A, T, A, right? And this can change to A, T, G, right? So that would be a mutation where we have a change in a single letter. Now, one of the really interesting things about our genetic code is that oftentimes when we just change a single letter, that does actually not change the amino acid that it codes for. So there's redundancy in the genetic code. So you might have noticed that sometimes you'll have a change and it is said to be a silent mutation. So for example, these both, if we looked at them, we could transcribe them, right? And this would transcribe to A pairs with U, T pairs with A, and this is U. And then this one would transcribe to U, A, C, right? And then both of these would translate into tyrosine. Right? So I just transcribed and then translated. I only know that it does because I just looked up on the table, okay? So this is what we would call a silent mutation. Meaning that the change in the genetic code has no change on the protein structure. And this is fortunate because if we have a mutation, a lot of them could just be silent, right? So my gene for hemoglobin might vary from your gene, but we produce the same hemoglobin molecule, so we cannot tell the difference. And these mutations that start to accumulate and are silent are oftentimes the basis for looking at variation between individuals, like in DNA fingerprinting and trying to determine the variation that's seen in the population as a whole. And then mutations can also accumulate in the part of the genetic code that doesn't code for anything, right? So we talked about how the DNA, there's only like 20,000 genes in our genome. So there's lots of genetic material that doesn't code for anything. And so if we have a mutation there, it's obviously not going to have much of an effect. So we start to accumulate mutations. So we could also have ATA changing to GA, okay? So notice how instead of the last letter, I actually changed the middle letter. And so for some reason, the last letter tends to be very variable. They call it the wobble effect. That you can change the last letter of a codon and it doesn't have any effect many times. But if you change the first or the second letter, then it can have a change. And so this would, ATA would be UAU, and that's tyrosine. We already discovered that. And then this would be UCU, right? And UCU codes for serine or SER, right? And so they call this a missense mutation. Sense mutation. Because now we have a change in the sequence of amino acids. And so that could very likely affect the way that the protein folds. And the way that the protein folds determines its function. So it could be like a defective enzyme or a defective protein in the plasma membrane, like in cystic fibrosis. Cystic fibrosis is due to a defective protein that sits in the plasma membrane and transports chloride ions in and out of the cells that line the lungs. Okay, so we can have ones. So the opposite of a silent mutation would be one that does have an effect on the phenotype, which is called a missense mutation. Yep. Any questions about that? Okay. So sometimes we can also have what are called frameshift mutations. And this is where we have an addition or deletion of a base. And this is causes a much more dramatic effect. So it causes more change in the protein than a substitution, than a point mutation causes. And the way that we can think about frameshift is actually using a sentence. And that makes sense. That's only composed of words that are 3 letters. So you could say something like, the cat ate the rat, right? So a point mutation would be like if we took this out and you replace the E with like an A, okay? So then it would read the, but with an A, cat ate the rat, and it didn't change much the meaning of the sentence, right? But let's say that we deleted the E, okay? Then the way that the machinery of the cell would read this is it would read it just using triplets. So it would go, right? And so that is complete nonsense, right? So notice how the difference between a substitution and a deletion. So you notice I just deleted that E, so I frame shifted everything over. So I just got rid of the E and then I'm reading it in groups of 3, and that's what it turns into. So that's a much worse mutation to have than, say, for example, a point mutation. The other one that I want to talk about is gene duplication. So this can sometimes happen if, for example, there's a mistake during crossing over, or there's a mistake, generally it's crossing over, but it can also be viruses, and it can also be due to transposons. So transposons can sometimes make copies of themselves and another gene and then place it into the genome. So this is where we get an extra copy of a gene. That can then be modified to perform a different function. So we have a lot of extra DNA, and some of it is due to gene duplication. So for some reason, the gene gets copied twice instead of once. And then all of a sudden we have extra genes, and it might start to accumulate mutations, and then it might become something that is important. So we're going to look at an example of that today in the video that we're going to watch that talks about the evolution of new genes, how we get new genes appearing in the genome of an organism that were not previously there, okay? Okay, so this is kind of describing like the different, structurally, the different types of mutations, but we also need to talk about the different types of mutations as it relates to how they are inherited and in what type of cells they are from, okay? So we can look at cell types and mutations, okay? So most of our body is made of somatic cells. Somatic cells are body cells. we accumulate mutations all the time in our somatic cells, right? So mutations, every time the cell reproduces, it could mutate, right? And it could eventually possibly become cancerous. But these are not inherited, so they are not passed on to the offspring. So if I get a mutation in a skin cell that then causes that cell to become cancerous, that mutation doesn't get passed to my offspring because it happened in my body, and that's not a cell that's going to give rise to my offspring, okay? So the difference between somatic cells and germline cells is really important. So germline, think this is your lineage. This is what you pass on to the next generation. So germline cells are the cells that give rise to eggs and sperm. Right. These are, so the genes in this, so mutations would be passed on to the offspring. So that is why, as I mentioned before, why they protect your ovaries and your testes when they give you x-rays, right? Because if they accidentally, you know, if something mutates in a liver cell, for example, that's not that big a deal if it's in one cell. But if you pass the mutation onto the offspring, every single cell of that offspring is going to have that mutation, and it can cause really bad effects, right? So it could be that the embryo is spontaneously aborted or that it has a bad defect in it. But it is also, these are the mutations that are important evolutionarily, right? Because these are the ones that get passed on to the next generation and might be important in future survival. So germline cells are the cells that the mutation, the germline mutations versus somatic mutations. So we write that. So somatic mutations, and these are germline mutations. Oh, we talked about gene duplication. Okay, so I want to talk a little bit more about genetic switches, because remember that I mentioned that this is another way that we regulate gene activity. So genetic switches are genes that can control gene expression. And this expression, this timing of when genetic switches are turned on and turned off can determine the differences between even species of organisms. So when genes are turned on and off, even embryonically, this can explain some of the differences we see between species. And so we're going to start talking about, between now and the end of the quarter, we're going to start talking about evolution and how important this variation is. We can see between species. Okay. So I'm going to show you a video that talks about a genetic switch in a fruit fly. And fruit flies are oftentimes used in genetic studies. In the 1920s, they started giving fruit flies x-rays, and they actually mutated them. And so some fruit flies, for example, got a mutation that made their wings all shriveled up, and some got mutations which made their eyes a different color. And so we were able to create all these mutant fruit flies, and they were subsequently really important in our understanding of how traits were passed from parent to offspring. So we used, Gregor Mendel used naturally occurring traits, but Thomas Morgan was the person that used artificially created mutants, fruit fly mutants, to study how traits were inherited. So we'll just watch this video. First, it talks about humans and our DNA, and then it goes on to talk about the variation between species due to these switches.
One might think that you can understand all of evolution simply by mapping the genes of every creature. Identify all the genes, identify all the differences, and you could explain the differences between, say, mouse and monkeys and humans.
So when the Human Genome Project began in 1990, the scientific world was on tenterhooks. All 3 billion letters of our DNA were going to be identified in order. In parallel, the DNA of some animals and plants was also being sequenced. Surely this would be a quantum leap in our understanding of how different life forms evolved. With this came another idea, that complex animals like us would have many more genes than simpler ones.
Now here we are, the most complex and sophisticated animal on the planet, right? You might think that would require a whole lot more genetic information.
The betting was on. Just how big would our genome be compared to other life forms?
There were estimates that humans would have between, let's say, 80,000 and 120,000 genes.
So when the final answer came in 2003, it was a shocker. 23,000 genes. The same number as a chicken. Less than an ear of corn.
I mean, people were freaked out by the relatively small number of genes. It's down to something like 22 or 23,000 protein-coding genes in the human genome. The simple nematode worm has about that same number. And there are plants that have considerably more genes than the glorious human genome.
The whole Human Genome Project has been a humbling experience as we've discovered that actually it doesn't take as many genes to make a human as we'd all hoped.
And it wasn't just that we had so few genes, but many of our key genes were identical to those of other animals. Huge though the breakthrough had been, the genetic revolution had opened up a whole new set of puzzles. As a solution to the mystery of how evolution works, genes and their mutations were only part of the story. There had to be something else, more subtle and more mysterious going on.
We have to explain then, how do you get all these differences if you have really similar sets of genes?
The quest to uncover what Darwin never knew would have to start again. The first tantalizing clues would come from those life forms that Darwin himself had studied, embryos. Look at these embryos. It is almost impossible to tell, just days after conception, which is the chicken, the turtle, the bat, the human. They look almost the same. Only as they grow does it become clear which is which. Darwin wondered, as scientists do today, how could they start out so similar and end up so different?
There is something profound about what the embryo is telling us, and we have rediscovered what Darwin was talking about all along, that the embryo is where the action is in terms of animal diversity. It is the platform for diversity.
What fascinates modern biologists is that all these different animals don't just look the same. They are using virtually the same set of key genes to build their bodies. The body plan genes determine where the head goes, where the limbs go, and what form they take, whether they are arms, legs, or wings. Another set of genes determines an animal's body patterning, the blotches, the stripes and spots. It is the same genes at work in every creature, from the leopard to the peacock to the fruit fly. And yet they produce radically different results. This has led scientists to a crucial insight about how animal bodies have evolved. It's not the number of genes that counts.
It's not the genes you have, but how you use them that generates the great diversity of the animal kingdom.
Finding out just how these same genes are used to create such amazing diversity has been the work of Sean Carroll and an unlikely hero of modern science, the fruit fly.
As much as I'd like to study the mammals of the African savanna, they make poor choices for laboratory animals. They're large, expensive, and reproduce very slowly. To get data, we have to find the simplest examples of the phenomenon we want to understand.
But the humble fruit fly does weird and wonderful things. This fruit fly is dancing for sex. A rapt female takes in the show. She's particularly besotted by the dark spots on the male's wings. Watching it all is an equally besotted Sean Carroll.
You might think them just to be annoying, but they're really charming. And the males of this species does a rather elaborate courtship dance where he displays these spotted wings in front of the female. To us, is as magnificent as what a peacock does.
But in some species of fruit fly, the males don't have wing spots.
So there's another fruit fly species that's different from the spotted species in two important ways. It doesn't have spots on its wings, and it does a lot less dancing.
Here then is a classic evolutionary puzzle. Why does one type of fly have spots and the other doesn't? Sean Carroll wanted to know. What is going on in their genes that makes them different?
So we wanted to take apart the genetic machinery for making wing spots to understand how those wing spots evolved.
Carroll began the process of sifting through the two types of flies' DNA. He had one clue to set him on his way. He already knew the gene that codes for the black wing spots. He calls it the paintbrush gene. But surprisingly, when he compared the genes of the two flies, they both had that gene, and yet only one had spots.
When we look at that gene in the two species, really, they both have this paintbrush gene. So the big difference is not having the gene, it's how they use it. One species uses it in the wing to make spots. The other one doesn't.
So why did the paintbrush gene create spots in one type of fly, but not in the other? In search of answers, Carroll turned to one of the least understood regions of DNA, the vast stretches that were once known as junk. It has been called the dark matter of the genome. Mysterious, uncharted, strange. The vast bulk of the double helix, some 98% of it, doesn't code for proteins, which make the stuff of our bodies. The genes which do comprise just 2%. Even now, no one is sure what much of this huge non-coding area actually does. but it has long beckoned evolutionary detectives like Sean Carroll.
So this is the Ben.
That's the fragment test.
Carroll had already learned that the paintbrush gene itself was identical in the two types of fly. So he extended his search through their DNA. And in one place, just outside the paintbrush gene, he found an important clue. A stretch of DNA that was different in the fly with wing spots. What could this mean? So Carroll conducted an experiment.
So you inject into the ferrets.
He decided to put that mysterious stretch of DNA that he found in the spotted fly in the unspotted fly. To help him see if it had any effect, he attached it to a gene from a jellyfish, a gene that codes for a protein that makes the jellyfish glow.
We cut the DNA up into little pieces, and we hook it up to a protein that glows in the dark. And then we inject that into the unspotted fly.
And then something remarkable happened.
When we looked at those unspotted flies, we see now their wings are glowing in the dark with spots.
Somehow, that mysterious stretch of DNA had turned on the paintbrush gene in the unspotted fly's wings. Once spotless, now it had luminous spots.
Bingo, we found the piece of DNA that mattered.
Carroll had found something that is revolutionizing our understanding of how different animal bodies have evolved. A piece of DNA called a switch. Switches are not genes. They don't make stuff like hair, cartilage, or muscle. But they turn on and off the genes that do.
Switches are very powerful parts of DNA because they allow animals to use genes in one place and not another, at one time and not another. And so choreograph the spots and stripes and splotches of animal bodies.
In the case of the fruit fly, it's a mutation, a change in just a few letters of the DNA that has caused the paintbrush gene to be switched on. And so a whole new species with wing spots has been created. But switches are now explaining far more than that. They are helping to solve many perplexing evolutionary questions, like how one creature can become another creature by losing its legs.
We'll stop there. Okay, so that was a mutation not in the gene, but it was a mutation in the switch. So if you get a mutation in the switch, then the gene can be turned off or turned on at different times or just permanently, right? So you can have mutations in switches that can then have effects on the body, okay? Are there any questions about that one? No? Okay, so I have the other video up, and this other video talks about an ice fish. And the ice fish has a gene duplication, and it also has a gene that is normally turned on in other fish and in us, but has been subsequently turned off. So you want to write down, you know, using these as examples. So if I asked you an example of a genetic switch, you would say there's a genetic switch that turns the paintbrush gene on or turns the paintbrush gene off, or in the case of the ice fish, it regulates hemoglobin.
For life to survive, it must adapt and readapt to an ever-changing Earth. New traits and capabilities must be invented, while some traits may be lost.
To see one of the most dramatic examples of how this occurs, we will follow a voyage of long ago that will take us 1,600 miles off the southern tip of Africa to one of the most remote places on the planet, Mutation. A speck at the edge of the Antarctic, it's pummeled by water so wild that getting there was no sure bet, especially in 1927, when this Norwegian steamship left port.
It's cold, it's windy, it's rough waters, and they're far away from civilization. So it was a really long and arduous voyage from Norway to eventually the shores of Mutation.
Norway had dispatched the expedition to claim the island as an outpost for its whalers. And after 2 months at sea, on December 1st, the crew finally rowed ashore and erected the Norwegian flag. But that's not the most important achievement of this expedition.
One aspect of the voyage was to understand all the sea life that was there. And so there was a zoologist on board, Ditlev Rustad. And Ditlev job was to really just throw a net over the side and trawl up, see what he found. But one day, in fact, the day after Christmas, 1927, he pulls up a really unusual looking fish.
A creature that many years later would illuminate our understanding of how evolution works. A creature so unexpected, it is still barely believable today when taken from a freezer at Northeastern University by a biologist who spent much of his career trying to make sense of it.
Well, Sean, it's easy to see why the fish was originally called a crocodile fish. Notice very big protruding jaw with lots of teeth.
Incredible, but it's so translucent. It seems like I can see inside the fish.
Well, the reason you can see inside the fish is that this is a scaleless fish. And in fact, if you, well, that's the brain. You can actually see the brain through the skull. And then you can also see, projecting from the brain, the optic nerves going over to the eyes. And Sean, for comparison, we can take a look at a cod. And when I pull back the gills of the crocodile fish,
Creamy white, look at that.
and here, if we take a look at the cod,
Oh, brilliant red.
yeah, very, very red.
So that would have surprised Rustad, because all the other fish he had seen would have red gills.
Absolutely.
But that wasn't the most incredible thing about Rustad's find. That came to light when Rustad, like any good Norwegian confronted with a strange fish, filleted it, as I'm doing today under Bill Dietrich's watchful eye.
Unbelievable. Everything's white, everything's colorless. You know, all the organs, all the soft tissues, the heart even. Every other fish he had opened in his life, it was full of red blood, but not this fish. And he wrote in his notebook, boldly,
Colorless blood, blue, farveless. The crocodile fish, or ice fish, as they've come to be known, had a dilute liquid almost as clear as ice water flowing through its veins.
okay,
And here's your ice fish blood.
that's amazing.
Except for the ice fish, all of the vertebrates on Earth, reptiles, birds, mammals, and all other fish, like this cod, have red blood coursing through their veins.
So what's so incredible about having no red blood cells at all is that, for example, our blood, 45% of our blood by volume is red blood cells. So we have enormous numbers of red blood cells in our blood, as do most other backbone animals. And when our red blood cell count decreases a little bit, we call that anemia. And if it decreases a lot, that's life-threatening. So it's stunning that an animal could get by with no red blood cells at all.
Why would a fish abandon a way of life that has nourished its ancestors for 500 million years? It has to do with the extreme habitat in which it lives.
The more cells that are present in blood, the thicker it becomes at cold temperatures.
So this watery blood allows the fish to live in really cold temperatures. So if it had blood like this, It'd be all gummed up.
That's correct.
All right. Blood is red because it contains hemoglobin, the protein that delivers oxygen throughout the body. Yet the ice fish had not one hemoglobin molecule. The fish absorb enough oxygen from the sea through their scaleless skin so that they can make do without hemoglobin. But the lack of red blood cells still doesn't fully explain how any blood flows in oceans this cold, below the freezing point of fresh water and almost at the freezing point of seawater.
And fish cannot live generally in that sort of environment because the freezing point of the blood is higher than the freezing point of seawater.
Yet fish thrive in these waters, ice fish and other species, all members of a group called the notothenioids. To show just how different Antarctic notothenioids are from other fish, husband and wife biologists Arthur DeVries and Christina Chang Dunkhorn in water as icy as the ocean outside their lab on Antarctica's McMurdo Sound. The temperature, minus 1.8 degrees Celsius, is so cold it's below the freezing point of fresh water and blind.
So as you can see, this fish is doing fine at this freezing temperature in the midst of ice crystals.
How these fish thrive in such cold waters baffled scientists until the 1960s, when Art DeVries found that Antarctic fish had invented something that protects them from freezing: antifreeze.
These fish have a certain protein, and what it does is bind to the ice crystal in the fish. And by binding to it and preventing it from growing any bigger, the fish doesn't freeze.
So only one group of fish in the Southern Ocean, the notothenioids, ice fish belong to that group, have antifreeze. So it's an invention unique to this group. And that's allowed those fish to invade a space that other fish couldn't live in. And these are nutrient-rich waters full of lots of food. So these fish are thriving now where other fish couldn't thrive.
Antifreeze proteins give notothenioids a clear edge where they live. But their existence poses an evolutionary mystery. When and how was antifreeze invented? The waters around Antarctica were once a temperate 10 degrees Celsius. But 34 million years ago, Drake Passage opened as Antarctica broke away from South America. Now a continuous current circled the new continent, isolating its surrounding waters. As a result, those waters chilled to minus 1.8 degrees Celsius. So notothenioids and antifreeze evolved in the last 34 million years. But traits are encoded by genes. Where did antifreeze genes come from? DeVries, Chang, and Lingbao Chen worked out part of the mystery in the 1990s by noticing that parts of the antifreeze gene, labeled in red, strongly resembled a different gene, that one gene gave rise to the other. The process began when the ancestral gene was accidentally duplicated. While one copy remained the same, the other accumulated mutations that eventually gave it a new function: to make antifreeze proteins.
The invention of antifreeze is a crystal clear example of inventing something new from something old, borrowing sort of the code of a pre-existing gene and then altering that to create a protein that has entirely new functions. And this story we see again and again in evolution, inventing something new from the old.
Notothenioids invented antifreeze genes, but one family, the ice fish, went a step further and eliminated red blood cells and hemoglobin altogether. As for how that happened, the answer has also been preserved in DNA. And Bill Dietrich found it when he compared globin genes from ice fish and other fish. The globin genes encode the hemoglobin protein.
So what do we see here?
Well, as you go from left to right here, the normal red-blooded globin gene is shown in the sequence on the top. Down below, we have the sequence of the ice fish. And as we move from the left, you can see there are dots. These are exact matches in the sequence. However, at the arrow, we see we get virtually no match. This is essentially genetic gibberish.
So it's broken right there.
That is exactly right.
At some point in history, a mutation wrecked this gene. And since hemoglobin was no longer necessary to ice fish, the mutation wasn't weeded out.
And this is the fossilized remnants of that gene.
So if the gene has become useless, it will eventually be lost as these mutations pile up and just erase what used to be there.
That is exactly right.
So genes are born and genes die as species find ways to survive in an ever-changing world.
This remarkable fish that Ditlev Röstad dredged up 80 years ago has taught us is that evolution doesn't always come up with the best solution imaginable. It just comes up with the best solution available. And that sometimes means getting rid of things that worked in our ancestors, as well as inventing new things like the antifreeze. And that record of changing habitats and changing lifestyles and changing genes is etched in the DNA record of life.
Okay, so let's write some notes about that. Okay, so the first genetic switch that we talked about, or that we saw in the video, was the switch that regulates the paintbrush gene. So you could put fruit flies. Okay, we have a switch that regulates paintbrush gene. Right? So in some species, the switch is mutated. So a mutated switch, gene is turned off. And so when they put the regular switch back in, and they would have done it embryonically, so they would have put it back in the embryo of the fruit fly, that fruit fly developed with that paintbrush gene that had the jellyfish gene and caused it to light up. Okay, so that's an example of a mutation that caused a change in the way that the organism looked. And then we have the ice fish. So in the ice fish, the hemoglobin gene was turned off. And so there was a mutation that turned that hemoglobin gene off. So maybe it was a mutation in a switch initially, right? And so that fish that did not have hemoglobin did better because their blood wasn't as thick, right? So they lost their red blood cells altogether. So they have no red blood cells in their body. So then the hemoglobin gene started gaining mutations. So it accumulated mutations over time. And so they actually referred to that in the video as being like a fossilized gene. So you could actually find one that was turned off that then started accumulating mutations. And it was able to accumulate mutations because even if a mutation happened, the gene was turned off and it had no relevant effect on the phenotype of the organism, okay? So this is what is referred to as a death of a gene, right? So the other one was the antifreeze protein, or the antifreeze gene. The antifreeze gene was a copy of another gene. It was actually a pancreatic enzyme gene called trypsinogen. A copy of another gene. Right? So we have gene duplication, which is now believed to be really important in creating variation, right? And then it accumulated mutations until it became functional. So it keeps the body fluid in the fish from freezing by binding to ice crystals. So it keeps, that's why it's called the antifreeze. So the protein that's produced by this gene. So this is what is referred to as the birth of a gene. Right? So we have a new gene that was not present before. That then has a dramatic influence on the phenotype and allows the organisms that have that gene to be better able to survive in those really ice cold waters. Okay. So now you have examples of different mutations having dramatic effects on the phenotype of an organism, like the presence of hemoglobin or the presence of an antifreeze gene or the presence of spots on the wings. Okay. Okay. So I'm going to stop there for today, and then we're going to talk about biotechnology next week. So we're going to talk about cloning and gene therapy and those kinds of things, and genetic transgenic organisms. Don't forget that you have a homework assignment due on Sunday.
The reason behind that.
I have another, oh. This one over here. So your clickers.